So there's a breakdown for the exam, guys. 10 true false questions, 58 multiple choice questions, and then 4 essays. 2 of them are going to be worth 10 points apiece, and a couple of them are worth 6 points apiece. Let's see here. The exam will be available for you to start taking at 5 o'clock on the 13th, which is Sunday. You can take it at 5 o'clock on 9/13 if you like. I don't have a time frame on it, so you will, once you sit and start to take it till it's finished. You can't just go away because you're taking it with responders. So, you know, as soon as you go and walk away, you're going to get flagged, and we're just going to assume you're cheating and just give you a 0. Save me some grading. You can take it. It is ultimately due by the end of the day, or by 11 a.m. on that Tuesday. We won't be having class that day. We won't meet for lecture, so you can take it during the class time. You can take it at 1 in the morning on a Monday morning. You could take it at 11 o'clock at night on a Monday night. I don't really care when you take it, as long as you sit down and take it, okay? So any questions about format, logistics for the test?
Will I have extended time on the test?
There is no time frame, so I don't know what extended time on no time frame is. So
I sent you an email. It's on email, the disability form.
Yeah, there's no time frame. If I had a time frame for everybody that said it had to get it done in 2 hours, then I would have set up for you to have the time and a half or the double time or whatever the thing would have been, the accommodation.
Oh, okay.
Yep, but I don't have any time frame. There is no time frame for anybody, so no time frame. So if it takes you 7 hours to take a test, then there you go. It takes you 7 hours. I will let you know if I do have a time frame. Okay. And when that's the case, I think there's maybe a couple people that have some time accommodations, and I will put that in the system as well for you. And it'll all be set up. But no time frame. Okay. You won't be able to get up. The system's not set up for you to go have a pee break or anything like that, I don't believe. Once you get up, you get up. So but any other questions about the breakdown, about when the test of it, you will receive an announcement. You should get an announcement on Sunday at 5 p.m. when the exam goes live. You can just click on that link and it will take you to it. And then you click on it and fingers crossed it works, okay? I will tell you this right now. If it is telling you that there is a password for you to input, I will tell you right now, there is no password from me, okay? I don't have a password protected exam that you have to fill in the blank or fill in the password slot there and start the test. Now, behind the scenes on the responder side, there is a password, but you don't need to know it. It is automatically put into the system and it automatically starts. If for some reason it's asking you for a password, close the browser, start it up again, okay? I have seen this problem with a handful of students so far in the other 2 classes. The common theme is it appears that they're using the Respondus lockdown browser that Respondus told you to download. I've been told that you can access the exam through Blackboard itself. You don't necessarily have to go through the Respondus download. I don't know what in the hell's going on, okay? I never had a problem with it until this semester, so I don't know why it's having a problem. But for those 6 people that had the issue, they shut down the browser, they started it back up, and it worked perfectly fine, okay? So if that's the case with you, just shut it down, all right? If after the 2nd time it's not working, send me an email, okay? Try to troubleshoot it with Blackboard help. But if that doesn't work, send me an email. We'll figure it out. Okay. Don't freak out. Right. Don't freak out. I don't know how long the test is going to take you. All right. For some people, it takes 30 minutes. For some people, it takes 2 hours. All right. If you take test a little longer, if it takes a little bit longer for you, that's fine. Just make sure you give yourself plenty of time. All right. Let's see here. What else logistic wise? Get an announcement. There'll be a link in that announcement. You can click on that and it takes you to it. Again, you'll be using Respondus. They may ask you for an ID to make sure you are the person taking it. If you feel that the government is watching you, you can just put a piece of tape over your address and your driver's license number. And as long as we see the name and the picture ID and your lovely face sitting right next to it, that's all we really need it for, to make sure that you are the person who is taking the exam. Okay. Let's see here. What else? From a logistics standpoint, I can't think of anything else on that regard. One of the questions that generally always gets asked is, well, what are some potential good short answer questions? Can anybody think of any? Anybody think of any potential good short answer questions or essay type questions for chapter 1 at all? What have you guys been studying?
Would the 3 types of bonds be a good short answer?
Sure, that would be a good one. What was that? Was that basic?
Chemical bonding? I just had it in my notes.
That was in chapter 2.
Yeah.
Okay, we had our 3 types of chemical bonds, right? That was in chapter 2. That would be a good potential short answer question. Okay, we had covalent, ionic, and hydrogen. Those are the 3 ones that we talked about. Being able to differentiate the 3 types, so telling me what each of them is, what goes on, sharing, unequal sharing, one takes from another, okay? Weak, singular type of bond, but collectively really strong. So being able to maybe explain those 3, that would be a good potential short answer question. Okay. What else? What else have you guys been studying? What else do you think is a good potential short answer question?
Maybe the 6 basic characteristics that we talked about first.
Nice. That was in chapter 1. Okay, we had 6 characteristics of life. Let's see here. What was the first one? Living things are organized. Here, let me. So we had 6 of those here. Okay. Maybe being able to list those and briefly give a description about what that is. That would be a good thing to be able to describe. So there are 6 of these things. I don't have them all listed down. Living things are organized. So when it comes to organization, we have basically what we've been starting with, right? Where we started to look at atoms and molecules and compounds and how molecules and compounds come together and give us cells and those little organelles within them, the cellular structure. Excuse me. We will build upon this and look at tissues. We'll look at a few tissues when we start looking at photosynthesis. We start talking about cellular respiration, how we make energy in a plant and an animal, respectively. Then we continue up to an organ, group of tissues working for a common goal, give you an organ. Different organs working for a common goal get you the organ system, and then all of those systems interacting give you the organism, right? So this would be a very good thing to be able to know, be able to explain. And we just get more and more population, community, ecosystem, biosphere type of thing. So each level we just get more and more organized. And on slide 10, 11, they explain those 2. The second one, life requires materials. Okay, you require stuff. You require energy to do stuff, to use those materials to make bigger structures. And we'll be talking about those concepts in the next unit. Living things maintain homeostasis, number 3. Everything has a little boundary that it functions optimally within. So we have a number of systems in the human body that help to maintain this homeostasis so you can run at peak efficiency. Living things respond to stimuli. If we can't respond to stimuli, we can't interact with the environment. And if we can't interact with the environment, how in the world are we going to be able to go out and get materials, food, to get energy from? Okay, wouldn't be able to. If you can't interact with the environment properly, you're dead and you die, and you can't pass on those genes. So living things need to respond to stimuli. You need to reproduce. You need to have sex. You need to have offspring. You need to develop that offspring. You need to develop to be able to produce viable offspring. And we'll talk about that in the 3rd unit. And then when we talk about evolution, we have to adapt, okay? We have to adapt. We have to have a spontaneous mutation. It can be a screw up in the read of the DNA. But that doesn't cause any negative side effects, but it does create a change and an adaptation. Is that adaptation advantageous? Okay, does it give me some kind of leg up on the competition? Then I can pass those on to the next generation. Okay, reproduce and develop. So those 6 things, you can interweave so much in with those. It makes a good short answer question. It obviously makes a great question to ask a buttload of various other multiple choice type questions off of as well. So if it's not a short answer question, good God, it is a great, fantastic, multiple choice type question. Where is my... Okay. So we got characteristics of life. What else do we got in chapter 1? What else do we got in chapter 1? Chapter 1 is a great kind of overview of everything, but there's something else that's inside that. What else kind of big picture wise was in chapter 1 that we talked about that would be a good potential short answer question?
What's the name of the basic structure and function of all living things?
Say that again.
What is the basic unit of structure and function of all living things?
Okay, and that would be what?
A cell.
Oh yeah. Okay, that is a cell. Obviously, we're going to probably do more with describing the cell and whatnot from chapter 4 type short answer questions. But what do we have there? I mean, a lot of organelle structure. So being able to describe some structure and function for organelles, that may be a good short answer question. FX is shorthand for function. Okay. Chapter 1, another big thing you should be able to describe would be the scientific method. Okay. Again, there's a number of great potential short answer questions. If you can answer them as a short answer question, you can answer anything on the exam. It could be true, false, multiple choice, fill in the blank. It could be literally any type of question. Okay. So if you've been able to just write these down on a random, excuse me, on a random sheet of paper, and you've been able to put all that out, it's freaking awesome. Scientific method, we got what, 5 parts to it? Yeah, 5 parts, 6 if you kind of go into scientific theories and principles / laws. But we have observation, hypothesis, predict, and give experimentation or make an experiment, collect the data, run the numbers, and then make your conclusion. All right, so being able to go into those 5 major components of the scientific method, that'd be a nice little potential short answer question as well. We have properties of water. Okay. So we had 2 great potential short answer questions from chapter 1, characteristics of life, scientific method. In the second one, we have chemical bonds. I realize I'm not sharing. Okay. Chapter 2, chemical bonds, ionic, covalent, hydrogen. We also have properties of water. That's a great potential short answer question. If I just skip ahead to chapter 4, oh, there's that. There's a number of great little bitty ones. And another good one, guys, would be being able to compare prokaryotic organisms from eukaryotic organisms. Prokaryotics, very basic, don't have a true nucleus. It's not a nuclear bound membrane structure.
I have a question real quick.
I'll give you an answer real quick. What do you got?
Is this document, like, are we able to see this somewhere, or?
I am making it as we speak.
Oh, that's nice. Thank you.
No, right?
Yeah.
And if you're nice, if you guys are really nice and participate, I'll throw it up on Blackboard.
I'll buy you a pizza.
Booyah! There we go. What else we got? What else we got? What else we got? Comparing the types of scopes. Something in the past that we've done more in lab class, but since we don't really have a true lab exam, and I know you're heartbroken, that would make a good potential short answer question here. Okay. What else we got? What's chapter 3 look like?
you could do comparing the different biomolecules.
Mm-hmm. Yes, ma'am. Oh my gosh. There's a lot of things that you can do in this. You can compare the difference between them. You know, what does that mean? What's their basic building block? Okay? That's what we're really talking about when we're talking about what makes them. Their basic component parts. Okay. You know, what makes them, examples of, functions of, those are all kind of, those would be kind of collectively in, you know, comparing the differences between them. You know, what their basic component is. Man, excuse me. You know, like a nucleotide is a phosphate, a pentose sugar, and a nitrogenous base. You know, knowing which ones are associated with DNA, RNA. Ooh, that's a good one. Okay. I'll write it here, and something that's commonly forgotten. ATP is basically a nucleotide. Okay, ATP is a nucleotide. Adenosine triphosphate. Adenosine is the nucleotide. Okay, so ATP is a nucleotide. We don't really think about it in terms of nucleotides. We usually think of just DNA and RNA. Excuse me. We usually just think in terms of DNA and RNA and comparing the difference between those two, which is pretty easy. What, you know, double helix, single stranded, uracil, thymine, with the respective nucleotides. But ATP is one as well. Okay, adenosine triphosphate, and adenosine is a nucleotide. Okay. I don't really remember. Levels of protein organization. ATP as a nucleotide is probably not worthy of a short answer. I just listed down there because usually nobody remembers that. Levels of protein organization. We had 4 different levels of organization there, depending upon the complexity of the protein structure. Primary, secondary, tertiary, quaternary, and not everybody goes to that 4th level. Okay, primary is 1, secondary 2, tertiary 3, quaternary is your 4th level of organization. Really complex. Most proteins don't get that complex. They usually will stop at that 3rd level. All right. But, you know, being able to maybe briefly describe those, that would be a good short answer question. So comparing the different biomolecules, levels of protein organization, comparing and contrasting DNA and RNA, you know, those make all fantastic questions. And you're going to see it after this unit. But all of these things that we're talking about here, all these great potential short answer questions, I mean, they have their hands in everything in biology. Water is involved. If we don't have enough water in us, we're dehydrated. If we don't have enough water, we die. Water is used in chemical reactions. Okay, it's going to be used to break down stuff through hydrolysis. We can form metabolic water through metabolism, through cellular respiration. So all of these things that we're looking at here, I'm going to see if I can make this a little bigger for me. Yeah. All of these things that we're seeing here, okay, Characteristics of life. I mean, I can weave this entire damn class in that one little question there. Scientific method. Chemical bonds. I mean, we're going to be talking about structures throughout this entire semester. Properties of water, biomolecules, we're looking at those. Protein organization, DNA, RNA, we're going to be looking at mitosis and meiosis and how the cells divide and how all these little organelles go to where they're supposed to go and how we make DNA. So all of these questions here, I mean, these are going to be things, guys, learn it now because it's going to make the rest of the semester so much dang easier for you. Trust me, okay? Trust me. Because we're going to start building upon these concepts that we are learning right here in the very, very, very beginning of this class. In the next unit, we have cellular respiration and photosynthesis. And those are, I believe we do, yeah. Metabolism, cellular respiration, photosynthesis, that's how we utilize what we consume on the daily to make energy. Okay, we obviously don't photosynthesize, but cellular respiration and metabolism, we utilize. So we're going to build upon some of these basic concepts here that we're talking about here in the beginning and apply them to a slightly different, more complex thing. So putting the time in now to get this stuff, guys, I mean, I promise, I'm not just asking these questions just to ask, okay? There's a whole buttload of information in this textbook that I could put in here that you probably would care 2 bits about, all right? This is the basic stuff with science. This is the basic building blocks for biology, for human anatomy, for human physiology, for human nutrition, okay? This right here is the basic for you on the daily, okay? So understanding these things, being able to explain all of these things, again, they are great potential short answer questions, all right? They are good potential short answer questions. Again, I'm not going to ask, 15 short answer questions. You're probably happy that I'm not either, okay? But literally, if you could answer all of these, just grab a sheet of paper and start writing. And if you can put all of the information down, and when you're studying for this, don't sit there and, make a short story out of it, just bullet points, okay? But the big words, okay? Especially, for the characteristics of life, just, one, bam, bam, and then 2 little words to describe it. But in your head, you're describing it, but, you don't have to write out a paper or anything for it. And then looking at that over and over and over again just makes it so much easier to recall. Put it away, come back, okay? Maybe you had to start looking at history class stuff, whatever, okay? Come back after a day, take a look at it, go over the same stuff again, okay? It forces your mind to try to recall that and pull that back, and that makes stronger connections for memories in that brain. Makes it easier to recall, hopefully easier to recall, okay? Is there anything on here, guys, that you would like me to go over? Is there anything not on here that you want me to go over? Because this class period is for you in your review, not just me kind of going back over the notes all over again. But if there's something you guys want to go over, even if it's one person, type it in the box, private message me, okay, within the chat box there. Let me know. So I want you guys to do well. It's the first exam, right? Everyone freaks out over the first exam. The great thing about this, guys, if you flunk the first exam, you have several other more exams. You have several other more chances of getting points this semester. So I'm just trying to say, just breathe and relax, okay? Just breathe and relax. I think I have it set up to where the entire exam gets released, not just one question at a time. So you'll be able to just scroll down and go to the next question, right? Just scroll back up and make sure you go back and answer that question, all right? But if you don't know it, go to the next one. Come back to it. Maybe there's another question later on that sparks a memory. All right. I know there's questions about the different biomolecules, carbs, lipids, proteins, okay, and the different types that we have: monosaccharides, disaccharides, polysaccharides. For lipids, we have triglycerides, waxes, steroids, and proteins are just proteins. So there's really no subclassification. Well, there is, but we didn't go into it. Nucleic acids, DNA, RNA, ATP, those are our three that we have there. Some of the common things screwed up on the exam for that. If I look at chapter 2, again, the bonds are kind of the big thing there, guys. Properties of water, those are the kind of the big things. Stuff that's commonly missed, though, atomic number, mass numbers, But again, those are more multiple choice type questions. There's a question based on chapter 1 when looking at, chapter 1, with looking at natural selection. Just know, I don't know, I can't remember off the top of my head because I made this before the weekend and the weekend was rough. Who's kind of the person who proposed the mechanism of natural selection? That'd be Charles Darwin. And what are some of the things that drive it, or fuel, I think is what I said, and that would be mutations. Mutations fuel changes, natural selection, okay? So that's probably, there may be a question, I may have a concept question or an application type question associated with that picture of the deer eating the fuzzy leaves. So there may be something like that asked, but not a short answer. Got a question here. Let's see. So we got a question here talking about, well, we need to identify what those monomers of the biomolecules look like. I don't. Easy answer, I don't remember. If it would be, let's see here. Get rid of that. Oh, let's see here. I know I didn't ask any of like these isomer, at least I don't have any pictures of isomers. Same chemical formula, same molecular formula, just a different shape, different arrangement of the atoms. Those are what isomers are. When it comes to, being able to pick it out of a lineup type of a question, excuse me. This is a typical glucose molecule right here, 6 carbon atoms. Could you pull out a glucose molecule and differentiate that from, try to see, from a fat molecule? a triglyceride. Okay. Glycerol molecule and 3 fatty acid chains. I mean, this looks completely different from a carbohydrate. I know I contemplated that. A phospholipid, that looks way different from a carbohydrate. I know I was debating putting some picture on it. I may have asked one in that regard, but it should be very, very obvious in that regard. Okay. This is what DNA and RNA look like. There's RNA single-stranded, DNA double helix. So I may have a question like that, multiple choice wise, but I didn't really include a whole heck of a lot of those. You're welcome, Maddie. Let's see here. What else do we got here? You should be able, guys, to go back and look at your exam scores. If you go click on My Grades, if you click on My Grades and then click on the name of the quiz, you should be able to go back and see it. Let me make sure that there is a stupid button hit. I've had some issues on it. I have it set up for you to go back and look as soon as it's past the due date. So for quiz 1 and quiz 2, you should be able to, but let me double check and make sure I didn't hit a button that I should have hit in there, okay? So I will go back and take a look at that for those that asked. Let's see here. Got a request to look at this. All right. So here are our acids and bases, right? The biggest thing to remember, is this going to work? There we go. Pen. Okay. Acids, guys, release hydrogen ions. Okay. And that's hydrogen ions right there. Okay. All acidic substances get rid of an extra hydrogen ion. Okay. Bases pick up that hydrogen ion, or they release hydroxide ions. They can do either one. Okay. Either take up the excess hydrogen ion or release the hydroxide ion. That's the first thing you need to remember. Okay. Acids give up a hydrogen ion, bases will pick it up. Okay. And that helps to maintain homeostasis. Bases are also substances that give up that hydroxide ion, that OH, okay, that OH minus. Okay. So that's the first thing you need to remember, a common thing missed. The next thing here is that pH scale is a logarithmic scale. Okay. So when we go from a pH 4 to a pH of 5, that is a logarithmic scale. That is a 10-fold change. Okay. So it's only 10 times, a pH of 4 is only 10 times more acidic. Okay. A pH of 10 compared to a pH of 8 is 100 times more acidic. 9, 10. 10 times 10, 100. Okay. This is what is deemed acidic, less than 7. Above 7.2 is basic or alkaline. Okay. 7.2. Okay. On up. Goes from all the way to 0 all the way to 14. Smaller numbers are acidic. The higher numbers are alkaline, basic. Okay? And neutral pH is where we like to kind of be slightly basic. Okay? And see, there's our human blood. Okay. Said before, if it drops below 7, stuff doesn't work properly. Okay. It's acidosis. Okay. Just a higher amount of acids within the blood. That's all that word means. Okay. If we get too alkaline, it's not good either. All right. But it is a logarithmic scale. So what does that mean? Every number we go up, it's a 10-fold change. So 5 to 7 is 100. 5 to 8 is 1,000. Okay, so on and so forth. That's the big thing to remember. I can't remember if I have you calculate that, but you know, it's in multiples of 10. So I think you can all just add a 0 or multiply by 10. Okay. If I have a question on there that requires you to use a calculator, I have a calculator built into that respondus, and you can multiply your little heart away on that. So I hope that helps with that logarithmic scale thing. Okay. What else we got? We got some good questions coming up here. What else? What else? What else? Let me see if I can figure out if I've hit all these stupid little buttons over here. So let me go here.
Well, there'll be pictures of the nucleus where we'll have to match which like part of the nucleus is it. I don't know if I said that right.
You did say that right. I'm going to hit this. I'm going to hit that. Click submit. I can't remember if I put those on there. If I did, it would just be identifying the structure at the end of the pointer. Okay? It would be that type of thing. So just kind of a general, nucleus, nuclear membrane, nuclear pores, mitochondria, rough ER, smooth ER, Golgi body. Those are some of the common things that would be easily askable for you to identify. Vesicles, lysosomes, peroxisomes, a little bit more difficult to ask from a picture. Okay. While we're sitting here, yes, I can go over that question. While we are sitting here as well, if somebody wants to log on to Blackboard to see if you can access your exams or those quizzes, again, just go into My Grades and then click on the name, okay? And you should be able to. There it is. Okay. So here are our 3 scopes, okay, that you're kind of, a potential short answer question was, can you describe these 3 types or differentiate between these 3 types here? We have a compound light microscope, which you can see right here. In the middle, we have our transmission electron microscope, where we transmit through the thing. And here we have our scanning electron scope on the far right here, where we just kind of scan. When you scan something, you're just looking at it you're scanning around, you're getting a cursory look here. With scanning electron microscope, you're just looking at the surface here. So one big difference with all of these, One is obviously the use of photons, light, or electrons. Electrons are much smaller than photons of light, so you can get really, 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 really close to that specimen there. And you can get a lot more magnification because you can get really closer. Okay? It also allows us to get very close to that object, so we are reducing that minimum distance. So we have greater resolution in electron microscopes. So compound light microscopes, you can only magnify it a little bit. Electrons, you can magnify it quite a bit because you can get really close. We have greater resolution. We have a much shorter distance between the scope and the specimen. Contrast is just the shading that we get with it. Again, when you look at this one, I mean, there's not a whole, when I say the word contrast, you should probably think of it in terms of detail, okay? I don't see a whole hell of a lot of detail when I look at this light microscope here. Again, I'm limited by physics, photons. I'm limited by my ability to get very close to that, object. So I can only have so much resolution. I can only have so much contrast, okay? You can add some things to it, but again, I'm limited by the photons of light here. So I don't see a whole heck of a lot of detail here. See a little whip-like flagellum, maybe some vacuoles here, some vacuole structures, some contractile vacuoles. But I mean, here I can get really close to that specimen and I can see a lot of detail here inside. I have tremendous resolution. I have a lot of great contrast here as well because I'm able to get really close and I can see some of the structures. Now, can I tell what in the hell some of those structures are? No, but I can add something to it and I can tag different isotopes and I can irradiate this thing and those things get picked up. Those proteins can get picked up within certain structures and I can see great detail. Here with my scanning electron microscope, you coat it with a metal and then you can bombard it and then you can see such detail. Here's that flagellum whipping around. This is that little flagellum right here, okay? This is it right here. All right, so you can see a lot of detail. You can see this looks like it's just kind of undulating around and moving, all right? It looks like a slug. And that don't look like a slug over here. So you can see a heck of a lot more detail. And when we talk about all of these here with scanning, that's what we talk about. Thin coat of metal. Get a much more three-dimensional image of that outside of it. Same thing here. Okay. Greater magnification, much greater resolution. Compound, I mean, we're limited by light. We can't get as close. Okay. So because of those limitations there, the amount of contrast, resolution, magnification, all of that is diminished here in this guy compared to these two. Okay. Much greater magnification, resolution, much better contrast compared to this guy over here at the light microscope. So good questions. Okay. What else we got?
What was the difference between the animal and plant cells?
Okay. So here we have our typical animal cell, right? And here we have our typical plant cell, right? Some of the major things that are different here. Let's see, where's mine? Do I have it? Sweet, it's working today. Some of the major things that a plant cell has that animals do not. Cell wall. Chloroplast. And the large central vacuole. These are three things that plant cells have that animals do not. That cell wall is very thick and rigid, and that's what gives the ability for plants to stand up. Okay, it creates this thing called turgor pressure, and that's this thing is filled with water. This large central vacuole is filled with water. Okay. And when we fill that up with water, it exerts pressure on everything. So it squishes all that stuff, all those little organelles, right? Squishes those organelles up against the plasma membrane, which pushes up against the cell wall, which causes that plant to stand on up. If for some reason it doesn't get a lot of water, then the water leaves. Okay, it leaves and goes into the cytoplasm. And that exerts less force on the plasma membrane, on the cell wall. And so what happens to a plant when you don't water it? It starts to wilt, right? It goes down a little bit, right? That's because that turgor pressure is lost because of lack of water and a few other things. We'll talk about that later. All right. And then obviously the major one, chloroplast. Chloroplast are the photosynthetic structure. It's what makes energy for the plant. That's why they're autotrophs. They make their own food. Okay. Now, you can see right here, it says plant cell anatomy at the top. Who do we have? Okay. There's a mitochondria here. All right. Mitochondria are not exclusive to an animal cell. Mitochondria are found within the plant cell. Okay. There will be a situation, we'll talk about this in the next unit, where we don't have access to cool enough temperatures to keep certain structures open. So we can use the chloroplast to make energy. So we'll do it at night without the presence of sunlight, obviously. So mitochondria are going to be involved with the plant cell in making it energy as well. There's a lot that goes into that. I get it. I just went up and over your head. But mitochondria are in plant cells, all right? Something that's commonly screwed up on an exam, whether it be this one or the next one, mitochondria are part of both animal and plant, right? Not just animal. So 1, 2, 3 things. Exclusive to plants. Okay.